Hi, I'm Brendan. My poster outlines our systematic assessment of a simple question. Does gene expression response to a particular ligand change in different cell types? We asked this question because we're interested in predicting how cells talk to each other from single-cell RNA-seq. And currently we can only hypothesize that cells may be interacting based on the expression of cognate ligand-receptor pairs in pairs of cell types. If we knew what changes in gene expression we'd expect to see if a particular ligand had been activated, or a particular receptor had been activated, we'd be able to improve these predictions. In fact, some methods already do this, making the assumption that transcriptional response to signaling is generalizable across cell types. We aim to test that assumption here. We used Connectivity Map, a pharmacogenomics resource that includes gene expression assays of 295 ligands in 9 cell lines. If you average gene expression change across all 9 cell lines, those genes responding consistently will have higher magnitude Z-scores than those responding differently in different cell lines. That's what we were doing in the figure top left. Only 1/5 of ligands assayed, as you can see in the heat map on the right, had any genes that showed consistent response. That's the rows of these heat maps. Or sorry, that's the columns of these heat maps. Are genes the rows of the ligands. 1 in 20 ligands caused consistent differential expression in more genes than we'd expect by chance alone, shown in red in the far right heat map. In other words, very few ligands cause consistent changes in gene expression across different cell types. However, if we do the same analysis after averaging Z-scores across samples within each cell type, 8 in 20 ligands, so nearly half, cause differential gene expression in more genes than we'd expect by chance in at least one cell type, consistent across different dosage and duration of ligand treatment. The box plot, so that was bottom right, the box plot bottom left is showing the distribution of p-values when testing against the hypothesis that ligands are causing changes in gene expression of more genes than we'd expect by chance when averaging across all cell lines in red, or within each cell line in green, or specific to each treatment condition in blue. As you can see from the box plot, robust to different FDR thresholds, the connectivity map data suggests that ligand response is consistent only within each cell type. Since there are limitations to the connectivity map assay, we've used a collection of individual microarray experiments to corroborate this finding. Rank correlation of ligand-mediated change in gene expression was significantly higher in independent assays from the same cell type than across cell types, as seen in the first figure, and overlap of significantly differentially expressed genes was also higher within the same cell type versus across cell types, and that was robust to the thresholds used to define significance. In conclusion, independent datasets confirm our finding that transcriptional response to ligand stimulus is consistent within, but not across, cell types. Come chat with me at the poster session to learn why this isn't all doom and gloom for cell-cell interaction prediction. And have a good day.